My name is Gunnar Kleemann. I run a data science company, Austin Capital Data. See if that's this my intro slide. And a little bit about me because it matters for this story. I'm a biologist, a PhD biologist, spent a lot of years in academia, analyzing data sources, right? And typically sort of high-end ivory tower stuff, right? So the longer I spent in academia, the more nuanced the data got, the deeper and bigger and broader the information got, and the more specialized the expert was required to actually interpret it, right? So this is the fundamental tension that we're working with. I always thought there was something wrong with that model, and I apologize to anybody who's an academic in the room. I can see there are probably some, but that's just the story. I can't change the story. So after that, I went to UC Berkeley to what's actually was the library school, and got the MIDS degree for data science, because I thought, even though I had been coding for 5 years or whatever, I realized that I just didn't have the chops to really do a super good job coding and doing the analytics that I wanted to do. So I went to that school to get that. And in that school, I didn't only get the ability to do the data science, but I also got an appreciation for the library sciences, because this is sort of the history of that school is library science. It's much more of a soft tech type of history. So you're going to see that woven into here, right? This sort of mission into my talk today. Let's see. What I'm going to do is I'm going to sort of introduce the mission, introduce my sort of vision for where I think open data should be going, and what I'm doing and my group is doing to push in that direction. And then sandwiched in between there is a personal story about how I think this can impact. It's a cancer story, but there are many other stories across all domains that just very closely match the same story. So I'll do that, and I'm gonna, if it's too much, I'm not sure, but I've got code in there as well. So I pulled out, all the code that I used to make this happen, and I'm gonna walk you through the different steps of building a system like this. Okay, so here's the challenge, right? Much of today's most important data is hard to access, utilize, and derive insight from. Now, that doesn't mean that it's not there. We're actually in a really unprecedented time when we've got, like in biology, it's omics data, right? You've got high throughput, robotically generated data profiling entire organisms times 1,000 or 10,000, right? If you're talking about yeast, you're talking about, many, many more. So this is, to me, I believe, like the human, the culture hasn't caught up with this revelation that this is actually there, right? There are experts who can understand this, but I don't believe there's a full appreciation for all that can happen with this information. Why not? Well, it comes down to data management, right? Collecting, cleaning, and organizing data. And from the time when I started, this has gotten quite a bit better. There's tools that allow us to do this in a high throughput way, but this is still a challenge. Analysis is a challenge, and I predict it's going to be a challenge going out into the future. I don't, now this is an opinion, right? I don't believe that all the analytical value has been culled from these data sets. Although if you talk to a pharma, right, and you say we're going to give them open source data on their servers, they'll say, well, we've gotten everything we need to know about pharma from those data sources. We need actual new data because we figured it all out already. I don't believe that, but that's my opinion. And then it's challenging for experts and non-experts alike to get any information out of these data sets, right? Let's see. So how do we deal with this? This is like the golden question, right? How do you get from a point where you've got this incredible gold mine of resource to a point where we've got an incredible gold mine of wisdom, right? There's a gap there, and that's the challenge that I want to claim that I have part of a solution for, and I'll show you what I mean. So I believe that knowledge graphs, I know knowledge graphs and graph analysis is old, right? And there's a long history of it in academics, and maybe, but I think, modern knowledge graphs can help us sort of bridge the gap between the data source and the human, right? And that's going to be a large part of the story. So what's the company vision? The company vision is sort of putting necessarily juxtaposing what data science gives us today and what data science I think should give us in the future. We know that, you know, that governments can leverage data science in very powerful ways to affect their populations. We know that there's, you know, sort of this dystopian reality that's unfolding where you've got, you know, large scale profiling of entire populations automatically, right? We know that corporations do similar things, right? And we also know that we're paying for them to do it by streaming all this onto our cell phone. So that's sort of a, you know, that's, we all know that. The biggest challenge to me is to make these data sources more useful to everybody. Now, I used to actually say, okay, you know, democratization of data, go forth and basically lower the bar, and we're going to actually be able to put data sources out there. People will make good decisions, right? That was my pitch from maybe even a year ago. I think it's naive now. And you can look at some of these like popular meme. Do you remember before the internet, it was thought that the cause of collective stupidity was the lack of access to information. Well, it turns out that if you have a lot of information, you can still be stupid, maybe even more stupid. Again, that's an opinion, but I think it's a widely shared one. There's this, which is an article, it's just a snapshot from our article on the New York Times, Free Speech in the Age of Disinformation, right? So we've got different types of information, maybe some information that's propaganda, some information that's actually not. But what's really funny is that somebody disputed this article talking about disinformation, and then we've got this information here where Jeffrey Bertel, the high-ranking priest, was resigned over data that was sold. Right, his data was collected, sold, and de-anonymized, and they figured out, you know, this Catholic newsletter figured out where he was, and shamed him. So that's actually real data, right? That's not disinformation, but it's also, you know, private data, right? That's been sold because he might have signed off on something. So the point to that was, I'm going to argue still that we should lower the bar, right? But lowering the bar is actually causing a lot of issues. I'm not sure if these were data scientists that did this, but in the article I read, it didn't have to be, right? It could be anybody who could do this now. You got a spreadsheet, you figure out where the guy's house is, you figure out his phone ID and track it back. Anybody can do this now, right? Because the bar's been lowered. So anyway, so here we go. I've got a number of data stories. I think the difference between just dumping data and dumping capacity into the collective conscious, or into the collective space, is storytelling. And maybe that's naive too. I don't know what I'm going to say next year. But we've got BioPredictor was my first project. I was looking for a biology job. I wanted to use machine learning to figure out which ones would IPO. Collaboration, we can pull collaboration networks right off PubMed. Some of the technologies I'm going to show you is what we're using to analyze the cancer graph. So, you know, there are 33 million papers in PubMed, and that's actually a record of behavior and collaboration. So we have a collaboration graph. Cancer graph is a subgraph right now for pancreatic cancer. I'll tell you more about that. But basically what this does is slices of those 33 million papers, we've sliced out the pancreatic cancer papers, right? And put that into a knowledge graph and found some, you know, pretty interesting things that might be useful to somebody who's dealing with cancer. My argument is that can rinse, repeat. Once you get that going, this process can be repeated for, you know, leukemia. It can be repeated for any type of cancer and other multiple sclerosis, whatever you want to learn about. There's a cocktail graph, which, you know, keep your eye on YouTube. We're going to put some videos out that involve some personalities from UC Berkeley drinking AI generated cocktails. Another thing that's really interesting to my staff is education graph. A lot of them have been pulled their kids out of school because of, you know, COVID, but they don't really know where to go with this homeschooling. So we've been building a homeschooling graph, and then virus graph, and that's another thing that runs very much along the lines of what I think cancer graphs should be, because not only are we dealing with COVID, but we're also dealing with weather pattern change, which is one of the reasons why we're probably seeing pandemics coming up, because we're being exposed to wild populations that we haven't been exposed before. So the point behind this graph is that we might need to know about other things besides coronaviruses, and maybe generally, right? Okay, cool. So this is the personal starting point for the cancer graph. That's my sister, Tracy. Um, she got a pancreatic cancer diagnosis when she was 47. And she was already in stage 5 at that point. Or she was in stage 4 and stage 4, yeah, within 5 months. So she underwent aggressive chemotherapy. And as a data scientist and as a biologist, right, I figured I could help her. Maybe, I don't know, do something, right? What would you like? Can I do an analysis? And this is not a uncommon request for somebody who's got a relative with cancer, especially if it's a relative who, if you've got a biological background, right? I've heard this before. Somebody says, well, find the best places for me to go, find the cure, right? That's a pretty tall order. And I don't believe that this is what that is, but I do believe that, I know what she went through. She'd ask the guy down the block, what should I do? She'd go on the internet. She was, there's the Rick Stevens oil, right? I don't know if these things are actually working, but you can ask the questions against reputable papers if you have access to them. But if you can't read them, if you don't have access to them, if it's too much of a lift to get to the point of understanding, then you can't, right? So there's actually a dynamic tension there. There's a barrier that's not a barrier anybody's assembled, right? It's a barrier. It's a knowledge barrier. It's a gap. It's because this thing has gotten so big, right? The data structures, and we haven't sort of, it hasn't come to a point where it's generally consumable, is what I'm saying. So, so what can we do, right? What can we do? And that's where this came from, this idea. Now, this is the issue, right, with COVID too, right? Biology is challenging and nuanced. It's not really a reasonable expectation to even expect somebody with a biological background to be able to address any biological question, right? This is a hard thing. Like, there's a long road to understanding any of these things in a nuanced way. There's experimental data. You can do experiments, you can read papers. I think you get the idea. But I do think that there's a problem with going forward, and it comes down to this. There's an idea that there's one person or a small group of people who are going to find the answer, right? And they're going to lead us in the direction. And I just think that this is an old idea. That might have been true when there was only a few people who could read, or a few people who could afford to go to college. But now we have a large, partially educated population, right? We have people who should be able to understand this and have opinions that are informed opinions, right? That would be very different from what I see sort of unfolding in general, right? You've got people with a lot of opinions, but they're not really particularly informed. And I mean, at least maybe this is naive, but at least to me, if I was to choose between having an informed opinion and having references I can trust, and having an opinion that's informed by references I can't trust, I'm going to choose the better, the higher quality references, just because I care about what I say. And I don't believe that people out there, I don't believe that that's I think people might, that's a good idea. I think people might like that idea. So that's my argument. Can we provide good information to people about cancer, right? Cancer in particular right now. So, so yeah, here's the pitch. I guess I already gave it. We do plenty of work supporting big money companies. I think data science should be supporting everybody, right? So what do you do to put this together? Like, we're going to stitch this together. If I say that we should have an open source option, and I'm not talking about software, although we're going to use software, right? But I'm talking about open source to the data, open access to the data. And I'm not talking about NIH, and I'm not talking about, going to a repository that's hard to understand. I'm talking about going to a place that's easy to understand, right? That's the open source part, at least to me. So you could read the literature. Pancreatic cancer yields 105,822 technical references. These are research papers. I remember when I was first in grad school, it took me a full 6 months to get through a paper, right? Not through one paper, but to get through a stack of papers and understand them. There was a period of 6 months when I I just couldn't understand the papers, and I'd have to keep on reading them. And eventually it clicked and I could get the papers. I could understand literature. So I think that this may be something that's more generally true. So you could sort by relevance. I mean, you might be the devil's advocate and say, okay, this already exists. It's called Google, right? It's called PubMed. You type in pancreatic cancer, you get all the references. But again, I'll argue that this is just too much. And maybe her doctor already read this, but I'm not sure. I know a lot of doctors, I don't think they read all the literature. So I really think that maybe, I'm not advocating that people should go in and tell their doctors how to treat them, but maybe you might find something in the literature your doctor hasn't. I don't know. Again, dubious. So what I did want to structure this, right? So at the iSchool, we learned that actually talking to the customer is one of the most important phases, right? We could go ahead and develop things that we think are really important for a long time, and maybe just waste a lot of effort. So I talked to Tracy. I said, "What do you need? What's your most important point that you wish you could address? Is there anything that you can't get at that you wish you could?" She said a couple of things. She's actually was really tough. Like she was able to deal with the chemotherapy to a point that nobody had been able to do it in that clinic. So she said, I'm pretty special. I've been able to deal with things. I need people to study me, like study my biology, because I'm so strong. That was her first one, but that's a funny thing about her. And then there's chemo's really hard to stick out. Chemo side effects are really tough, right? So can I learn about what the chemotherapy is? What's going on with that chemotherapy? So I anchored on this question because it's a bit more of a easy one. But I do think that a very close second would be pulling references and finding out, who's studying pancreatic cancer, who's got the best publications, who's got the best collective network, right? Those are questions that we could address with this tool, and you could just put it on a map, right, and allow people to link back to the literature. Okay, watch my time. So here's the technical stack. Basically, we use Biopython. Biopython gives us an API we can use to pull publication metadata. So we're at the granularity of metadata here. I pulled 306 publications so the tool right now is able to get specific queries into for PubMed and then pull all the metadata from those papers, right? It's got a very limited scope: keywords, abstract text, authors, chemicals, and keywords again. And then we stuff this into something called Kraken AI. That's the old name for it. It's now called TypeDB, and I'll talk to you more about that. That's the main inference engine and storage engine that we're using. And then there's my company doing the interpretation. That's Austin Capital Data, and there's everybody else, the world, right? So that's the basic stack. So I'll just go into a little bit of the code. So we have some code in here. You can see Kraken is now Vatical. So if you really want to build something like this, you can go to Vatical, github.com Vatical to join them. It's a very good open source community, and they will walk you through every step of the way to get you up and started. Some of the syntax is a little different because this is the older version. This is the Kraken version. So you'll see a Kraken syntax here, but the ideas are the same. And if you were going to use this syntax, you would use Kraken 1.8. So we start the server. You could get the status. You're going to build basically a key space with a schema, right? So we're going to have a specific schema. We stuff it in there. Call this PubMed Ref Cancer. We can use brew to install this. So these are the installs. You can also basically just pull the image down and run it. For an easier avenue, you can actually leverage Banj the Man. He made Kraken Codex, which is sort of it's a Dockerized version of Kraken that has inserts and stuff like that. It's even got a front end on it, so you can, if you don't want to invest too much time in putting all the pieces together, you can pull this thing down and run it. All right. So the structure of the language is there's entities, relations, and attributes. Three main types of things. Types have behaviors. The attribute has a has. The entities can play roles, and then the roles relate things, right? And then you can calculate at query time, you can calculate rules. So you can make synthetic paths through the graph based on your rules. Let's see. All right, so design the schema. This is just to tell you sort of how we're putting this information together. It's important to think about the schema. So we've got attributes would be abstract text would have a sub attribute, right? Author name, these are PubMed article, author, PubMed ID, title, text, right? Those are all the different attributes that we'll pull out, the sort of the metadata. You can see we've got PubMed article sub-entity. So the entity structure is like this. So if we look at an article, it's got an ID, a title, an abstract text, a publication date, citation, author, right? And notice that these are playing roles and these are hases, so they're attributes. And then there's a key, there's like a unique PubMed ID, right, for each of those things. And then the author would be a sub-entity. They're both sub-entities, but the author has a name, a unique name, an affiliation, and then they play contained author. They play an author that's contained in a paper, right? And then the keyword similarly. Let's see what's next. So we could see the relation. So basically, contained author relates an author container to a contained author, right? So the author's contained in the paper and the paper is the container of the author, right? So that's sort of the schema. And you can see a similar relation for a keyword, for the keyword. All right. And then what we do is we load it up. You can use, this is the Grackle language. So match variable X, a thing, and get all of the things, right? So it's a very basic query showing us that the schema has been loaded there. This is just setting up the folder structure. I'm not going to bore you with that too much, but here's where we get to where the rubber hits the road. So here's the knowledge structure that we can build out for a specific set of relations that can be recursed to pull in all those 306 papers and make a much bigger knowledge graph. So we've got the PubMed article. It's got is a PubMed article. It's got a citation, a publication date, all the metadata for that PubMed article. Then we've got keyword, author, mesh term, journal, and chemical, right? So the chemicals are important for this question. The journals might be important for reputation or for other things. And then the authors, the authors are important, obviously. Yeah. Let's see. So pull the data from Biopython. So this is how you would query Biopython, right? So it's the API. You can see Graken mod search term and return 500 max. It's just the API syntax for BioGraken. I think I put it in here because it wasn't obvious from the documentation or something. It was like I had to go through a lot to find out how to write it exactly. But it works, it's robust. And then you can see basically what you do is you get back a list of papers. These are PubMed IDs. So we can stack up these papers and start to load them into our database. And then this is the basic query anatomy. Again, this determines what we can do, right? So that type QL is the name of or type DB is the name of the Graken now. P is a person, has a name Bob, has a phone number, and the phone number variable, right? So this is the basic syntax, and I can just show you some of those queries. Let's see. Maybe I don't show you that right here. Okay, let's jump back into the overview, right? So here's the basic pipeline. Take Biopython, pull in your publications, pull in your metadata, load it into the graph engine. And then the analyst is going to be able to serve this to the world as specific queries. Like I think what we'll do first is we'll do these canned queries, take suggestions from the public, and then serve those specific graphs for perusal, right? And then ask for more canned queries. So what do we do? 306 papers is still too large, right? So how do we make sense of 306 papers for anybody? How do we get any benefit from that? So the question again, just to remind you, are what are the most important treatments? What are the most important chemicals? what are the most important ideas? You might want to ask, like, what are the most important keywords? And then who can we contact about these experimental treatments? So those are the basic questions that were asked. You can see a chemo side effect query, right? So this is the query that would go into a graph to get the chemo side effects. Let's go back to that. You'd see match P is the PubMed article, has title T, has PubMed ID, right? And then we have the semicolon, and chemical is a chemical, has chemical name, and then chemical content. So this is the relationship, right? So there's a relationship here. It's a chemical from a paper, right? So that's the and then get the chemical for the chemical from the chemical container, and then limit 500. This offset is going to tell you how many, if you want to jump through to the different responses. So this is basically how you would query the graph that I showed that we loaded of those 306, and we can query it in various different ways. And the question here is, show us the chemicals mentioned in those papers. What are the chemicals mentioned in the side effect papers? We can think about this again. What are the most important treatments? And that comes down to chemicals. Which chemicals are connected and which are the most central, right? So what's being used commonly? What are the things that people think about these chemicals? Are there interactions that we should know about? Are there treatment that nobody has thought about, or that are only in a few papers? That's the other side of this, right? You might want to look at what's common. You might want to also look at what's not common. And then are there key combinations? So we start looking at cocktails. Are there cocktails of chemicals? Are there any indications that might match, like my sister, for example, for her situation, right? The side effects was really the question, though, because that was what she asked me. Symptom management ideas. What are the most important ideas and keywords? So how do you manage symptoms? Like, let's say there's a PubMed article that shows that these are the common concoctions, but are there any suggestions about managing symptoms, right? Can we pull that out of the data? Are there any, I mean, I don't know, just chamomile or something, right? Just peppermint, whatever. You can hear this in the popular literature, but can we actually get a real study that's actually got real information? So let's see, are there certain groups of papers that center around these ideas? Again, we can get to something that's very actionable. If you find that there's a certain, maybe there's a certain academic cluster, maybe there's a certain professor and graduate students, maybe they have this idea. Maybe you're very interested in this idea. It wouldn't be too much of a stretch if you have really studied this to actually write them an email, right? Once somebody's looked, as again, for the academics in the audience, once somebody's looked in detail at your work and they have something useful to say, it's not too hard to get your ear, I think. So I think that this is also really interesting. If you can identify communities that have ideas that are like your ideas, this is another thing that would simply wouldn't be possible, right, without this graph. So which are the most relevant for chemo symptom mediation, right? Which ideas are the most relevant? We'll go back. This is the basic schema. Now this schema can become much more nuanced, much more detailed. Actually, I work pretty closely with this company, and they're very much on the other side of this, right? I'm coming from a traditional training. Just basically, let's take the minimum and get the most out of it. And then the other side of this is, let's stuff everything that we can think of, right? Let's atomize every concept into this incredible hairball, and we'll get more value from that. So they spend a lot of time, engineering databases that are highly performant to do that kind of a thing. But that's a side note. Here's what you get, right? So when you actually put in 306 papers and pull out the chemicals, the pancreatic cancer chemicals, chemotherapy agents, and side effects, right? So you put those queries in. What are the chemicals to get this hairball? So what do you do? What do you guys think? What's the? I don't know. I'm curious what you have to say. This is the problem, right? This is the problem with knowledge graphs. I don't want to. So we don't know what to do, right? This is essentially like from Euler's time, right? You say, oh, this will be really great, and you get in there and you put all that information in there, and that's modest. That's modest. Well, turns out if you stare at it enough, you can actually pull some structure out, right? So this is just me going in and moving things around. And it turns out that there's a huge cluster right here of two chemotherapeutic agents that are used over and over again. This third cluster, I think, is a keyword for chemotherapeutic agent, right? So that comes up quite a bit. But there's actually two chemicals, and I didn't know about this, and I'm pretty sure she didn't know about this. So it really gets us very deep into the biology once we start to peel back, like what are those things. And you can see the chemicals are purple and the PubMed articles are red, right? So what we've got there are two chemicals that are highly cited, bound by a number of papers. Now, the other parts of the graph are also interesting, which just basically the depending on your polarity of your interest, right? Do you want rare stuff or do you want common stuff? So, and that would be up to the user. But what we see is we've got gemcitabine and deoxycytidine. These are two chemotherapeutic agents. And this is actually the goal. This is my goal anyway. But I always want to hear what other people think about what the goal should be. But my goal is to get somebody on their desktop with this essentially very nuanced and important toy. And I want them to say, oh, well, what is gemcitabine? Am I putting it in my veins? Is that what's making me sick? What is deoxycytidine? Is that something that I could use instead? Well, what is it? Like that spark is really, I'm also a teacher. So that spark is really what I'm after. I'm after somebody just wanting to know. And I think that the job is actually essentially done at that point. I mean, I want them to go back to the graph, but that's the main job, the main task. You raising your hand? Yeah.
Based on the way the paper that reference similar to it, it's making this is like general that
Uh-huh. Uh-huh. Yeah. Yeah, yeah. So the schema is very lean, right? So schema doesn't include references. So let's go back to it, just so I don't make a statement that's not true. I'll just go back to the exact schema, right? So it actually is just keywords, articles. So the article is the main container thing that binds them all together, and chemicals. So what you're seeing right there is we've got an article bound to a chemical, and that article is also if it's bound to another chemical on the other side, it's just the same connection repeated, right? That's actually what you're seeing. So it's not anything about the reference structure. It's that the PubMed article mentioned both chemicals. Yeah. Oh, I see what you're saying. They might mention another. This paper talked about that chemical. Yeah, yeah, yeah. Yeah, yeah, and it doesn't make the just. Yeah, yeah. No, that's it doesn't. That's not far-fetched to talk about context, but this is much more basic than that. Yeah, it doesn't make the distinction. It could reference another author that's talking about that chemical, or it could talk about both chemicals in tandem, and it wouldn't make the distinction at this point. Right, right. Yeah, yeah. So there's a named entity recognition layer to this. It's not included in this analysis, but BioGraken, which is the larger project, has that. So you could get into those nuanced, depending on your use case, right? Yeah, this is made to be lean and just to get people hooked. And because we don't want something that's overburdensome. But I mean, certainly for the much more the much more, I guess, professional questions, I think it might be much, useful for that. Yeah. Um, yeah, cool. Um, yeah, and this it can be easily swapped out. Once you have the infrastructure, you just change you just change the architecture and then build the loaders, and you've got your custom version, right? Um, okay, I think let's move. So what I did was quickly, reference the literature and look at what these things are. Turns out that they're very similar chemicals. Uh, One of them, gemcitabine, I think is associated with a lot of toxicity. So this may be the thing that's causing problems. And you can see that they're analogs, right? You can see that the structure is very similar right here. And let's see. You can see that they're also antagonistic to each other from this paper, right? So the structure is really similar. Deoxycidity and release from pancreatic stellate cells promotes gemcitabine resistance, right? So actually the body it looks like the body can release deoxycidine and antagonize the chemotherapeutic agent. So another piece of information that might be useful. Now, I'm not a cancer biologist. I have people who I want to bring in who are cancer biologists on this project, but this is new to me, right? So this information. Okay, so here's more code. This is basically how you would run this if you wanted to build it. I don't know if we would need to go into that in detail. Yeah, this is basically how we would pull out the API response here. And then basically build out the structure. So all those details are in there. I don't want to bore you with this. This is the insertion code right here. So yeah, basically use Python and gRPC and Kraken for the insertions. More code. All right. So what are the next steps, right? I think, having an open source front end, allowing downloads of these graphs and the data, maybe a more enriched data source depending on the use case. I think getting community feedback is really useful just to build these things out. And then looking at how this changes over time, I think would be really interesting, just to see how the state of the art changes over time. So you get a query that's just executed, every 6 months or every 2 months, and then we can look at how the state of the art changes over time. But I really a big focus of this is actually pulling in more brains, right? Pulling in more brains, pulling more diverse perspectives, maybe not just having cancer biologists, but having a place that other people who have other concerns and other perspectives can feed back and record their ideas. Now that's really tricky because it could, we want high quality thought, right? So this is really up in the air. And again, I'd like to hear more if you have ideas. So again, maybe an intuitive front, a drill down. I think accounts with memory makes sense. And then obviously the first step will be repeating this for multiple cancer types for anybody who wants to, who has cancer, who has a relative with cancer. You can actually look up like a quick question with my sister was like, does do massive amounts of THC cure cancer, right? I mean, that would be a really popular query, I think. So anyway, yeah. So we got 5 minutes. I'm just going to talk about a little bit more about what's out there in this in terms of this. TypeDB and BioGraken is an open source tool. So if you're interested in this, you can actually pull their loaders and get a large part of biology loaded onto your computer. So using BioGraken. And then you can stitch in whatever other loaders you can copy. We just copy the pattern, right? And you can stitch in new loaders. This is the BioGraken schema. You can see how different it is from what I have. This one has the named entity recognition layer for COVID data, right? So yeah, let's look at the data sources. So what it has right now, just as a starter kit, essentially, is precision medicine. We've got clinicaltrials.gov. It's got ClinVar for your clinical variations, genetic variations, CT database, DisGenNet for disease classifications, drugs at FDA, HGNC for disease or for gene names, Pharma GKB for pharma data. It's got PubMed as well for text mining. That's what we were referring to. Uniprot, so and that would be text mining over here. And then disease network information. It's got protein data, protein interaction data from Uniprot, Reactome, gglib, DisGenNet, HPA tissue, EBI intact, CanCode, gene expression, omnibus, and TissueNet. So there's quite a bit of information here. One thing I'd like to do is stitch these graphs into there. This over here would be, I don't know if it's obvious, but these are research entities. So this would be, you can pull out, where things are happening, who's looking at which protein, that kind of stuff from this data set. Organization is one of the schema items. Company is another schema item. Institution, laboratory, right? And they all have names, right? So there's that. You can patent owners, right? Patent applicant, that's in there. If you look at publications, right, that's over here. So here's PubMed with all its features and how it connects to proteins and the other like biological entities. So you can see this patent, I guess, over here is a publication here. And then biological entities like proteins. See if we have a zoom in, right? Protein and gene, chemical. So we've got that as well. And that's not the data, that's just the schema, right? So you can imagine, you saw how that simple schema turned into a hairball. This turns more into hairball. These are my people. They are mostly Austinites and Berkeley people. So these are people, all the brilliant people I met at school and locally where I live in Austin. So it wouldn't be possible without them. Yeah. And if you're interested in more of this, you can certainly talk to me. Here are some web addresses. This is our company. These are two websites that we're building. One of them I think says 404 right now, so don't go there yet, but like tomorrow. And then, sorry, it's a little tricky. Vatical, you can join the Vatical community if you want. They're great as well. And that's Kraken/Vatical/TypeDB, AKA TypeDB. And that's it.